Hello, my name is Daniel Mahecha. I am presenting our poster Enhancing Genetic Diagnostics, a machine learning approach to prioritize variants of uncertain significance, developed with Aidemar Núñez, María Claudia Latic, and Jorge Duitama at Universidad de los Andes, Colombia. The high-throughput sequencing technologies have increased the yield of variants that can be detected on a single test. Despite that, due to the complexity of the variant interpretation process, there is a high number of variants of uncertain significance. This problem is aggravated in underrepresented populations, such as the Latin American one, which lack the genomic resources necessary to achieve a more informative classification of variants. Here, we present a model to prioritize which VUS among all consequence types are suitable candidates for functional studies. In this graph, we can see the distribution of scores or of commonly used computer classification tools. SIG, for example, has an overrepresentation of pathogenic range scores for benign variants. Polyphen has a slight overrepresentation of benign scores for pathogenic variants. Rebel has a more uniform distribution with peaks on the benign and pathogenic scores, respectively. For VUS, we can see a double peak in Polyphen, while the Rebel scores have a more uniform distribution, but with a skew towards the benign range. Our goal was to train machine learning models to classify the VUS on a benign and a pathogenic category. The benign category is a merge of the benign and likely benign category of the ACMG classification system, while the pathogenic category is a merge of the likely pathogenic and pathogenic categories. To train the models, we selected about 80,000 variants from ClinVar, which were not classified as VUS and had at least 2 quality stars. To build the models, we selected a subset of about 75,000 variants. The models we chose to train were a random forest, a support vector machine, a multi-layer perceptron, and we optimized the hyperparameters, taking into account the area under the ROC curves. Additionally, we selected a subset of variants which were formerly VUS but had been reclassified as pathogenic/likely pathogenic or benign/likely benign. To train the models, we used as features 9 conservation scores, the LOF tool, the consequence type, and the allele frequencies. Our 3 models showed a greater performance than other tools for the whole XVUS test set. In particular, the random forest and the MLP had AUCs of 0.98 and the SVM of 0.99. This set is composed of about a third of missense variants, a third of synonymous variants, and a third of other consequence types. Across many different consequence types, our models have a higher performance as well, except on synonymous and indel, where the performance was not as high as the other consequence types. The random forest models yields an information parameter that lets us know the relative importance of each feature on the decision. We can see that the allele frequencies and the CIDD scores were very important in all the consequence types, although the allele frequency is more important on missense variants. Interestingly, the LOF tool is an important score for the classification of synonymous variants. Finally, we applied our models on a set of VUS, finding that most of them yield a score of pathogenic. We can see a peak in the pathogenic score on the 3 models, and a lesser peak or near the benign score, especially on the RF and the SPM. In conclusion, our approach can potentially help resolve the high proportion of VOS in our population. The models developed in this work can be useful for researchers working with variant interpretation and genetic-based diagnosis. The RF model is available at our GitHub repository. We are currently developing a new iteration that includes a set of pre-calculated scores for enhanced usability. We acknowledge the contributions of Sigen, Fabián Heredia, and Yacir Ramírez. Thanks for your attention.